Hi and welcome to my OCR A-level biology revision session with me, Christine. And today's lesson we're going to look at enzyme action. So taking you back to GCSE chemistry then, you know that molecules have kinetic energy, that they are constantly moving around and they are colliding. Well, what we need to understand is that for a chemical reaction to occur, those molecules must collide in the correct way and with sufficient energy. Now, this is what's known as the activation energy. The activation energy because it's what's needed to start the reaction, to activate it to happen. So what we end up with is our reactants are then converted into the product if that activation energy is reached. If it's not, then the reaction does not occur. So to understand enzymes then, we need to take a step back and look at our protein synthesis. We know that our genes are the genetic code for protein synthesis. So the DNA, the base sequence of the DNA, gets transcribed onto our single-stranded mRNA. That mRNA is made up of the triplet code, which is known as the codon, and each codon is translated into the amino acid which makes up the polypeptide. So we end up with our primary structure, our sequence of amino acids that are held together by this peptide bond. So the protein, once the peptide bonds have been formed, once we have our polypeptide, that will start to fold. And the secondary structure is whether it's the alpha helix or the beta pleated sheet. So that's to do with the weak hydrogen bonds causing this either alpha helix shape or the beta pleated sheet. Then it will continue to fold into what's known as the tertiary structure. So the folding, the bending of this protein, because of these intermolecular bonds, these interactions, so we're talking about our ionic bonding, we're talking about more hydrogen bonds, the hydrophobic, hydrophilic interactions, or our disulfide bridges. So our polypeptide is being converted and folded and bent in a way to give it its 3D shape, making it a globular protein. So enzymes are globular proteins, and they are globular proteins that act as biological catalysts. Now, a biological catalyst is something that's going to speed up the rate in which the reaction occurs. And they do that by forming temporary bonds with the substrate. And when they form these temporary bonds with the substrate, what that will do is that will put a strain on the bonds within the substrate, therefore making it easier for it to turn into, be converted into that product. So as they come together, what you get is what's known as an enzyme substrate complex forming. So the enzyme and the substrate will collide. When there is a successful collision, that will result in this enzyme substrate complex forming. That will then reduce the activation energy because of these temporary bonds that have been formed with the substrate, is going to reduce that activation energy that's required to start the reaction. So as you can quite clearly see, my energy reaction pathway graph has been reduced when there is an enzyme present. So reactions will happen naturally, but they need to have the right temperature, they need to have the right pressure, they need to have the right pH for it to occur at the right point. Whereas by having the presence of enzymes, this works in a way which we can speed up the rate of reaction. So inside the cells, these biological catalysts can speed up those reactions, ensuring that the products that are needed in the reaction pathway are produced at a sufficient rate. So enzymes can work in one of two ways. They can either do what's known as an anabolic reaction, where they're going to build something up. So we have our enzyme and our substrates, which will collide. When we have that successful collision, we end up with an enzyme substrate complex. The enzyme substrate complex will then be converted into an enzyme product complex after a condensation reaction has happened. So the condensation reaction is this joining of molecules to form a larger molecule. Now remember, because it's condensation, there is a loss of water as a product. So if we were talking about our saccharides, the glycosidic bond is formed through a condensation reaction, therefore water is lost as a product. If we're talking about our polypeptides, our amino acids are joined together by a peptide bond through the condensation reaction, and water is lost as a product. If we're talking about our lipids, it's our ester bond. So a condensation reaction is where we are joining monomers together to form macromolecules, and those macromolecules are not the only products. Water is also a product as well because it is a condensation reaction. So if it's not a case of building it up, we may be breaking it down. If it's breaking it down, again, we have our substrate and our enzyme. When they come together, we have an enzyme substrate complex. Now, hydrolysis is a reaction where you need that addition of water. The addition of water, lysis to split, hydro, water. So we're going to split by adding in water, therefore breaking our macromolecules into our smaller molecules, potentially our monomers. So enzyme action then, when we first discovered and understood enzymes and how they worked, we used what was known as the lock and key hypothesis. This is where we know that the shape of the substrate and the enzyme's active site are what's known as complementary to each other. Now remember, enzymes are polypeptides that have a tertiary structure, which means that they fold and bend in such a way that they have a very specific shape. So the part of the enzyme where the substrate is going to bind is known as the active site. So therefore, with the lock and key hypothesis, what we're saying is the enzyme is very specific because the shape of the active site is complementary to the substrate. They fit together like a lock and a key. Well, as we have learned more and we've done more experiments, we have investigated more, what's happened is we've advanced our knowledge now to believe what's known as the induced fit model. And the induced fit model means that the active site actually changes its shape slightly. So as the substrate comes and collides with the enzyme, because of these weak interactions, they actually cause the tertiary structure of the enzyme to change, and that is going to strengthen the binding between the enzyme and the substrate, and that is therefore going to put more strain on the substrate, therefore reducing that activation energy. So we know the lock and key hypothesis, one key, one lock. We know the induced fit model where the enzyme changes its tertiary structure, it molds itself around the substrate. So we know that enzymes are biological catalysts, and we know that they are there to speed up the rate of reaction. Well, some enzymes don't actually function without a little help. And in this case, if an enzyme needs a helper, and this helper is normally non-protein based, the enzyme is made up of proteins. This is a non-protein helper that is known as a cofactor. So some enzymes don't just have the right shape, they need a helper to ensure that they do have the right shape. And amylase is one of those examples. The cofactors can be inorganic. So in the case of amylase, amylase is an enzyme that breaks down saccharides, in this case, the polysaccharide starch. So amylase is going to hydrolyze starch down into maltose. Well, what the amylase is going to do is the amylase needs first a inorganic cofactor in the case of chloride ions. So when a chloride ion is attached temporarily, it is going to form the correct active site shape for amylase to break the glycosidic bonds in the starch, to break the polysaccharide down into smaller saccharides. Now, amylase and chloride ions is one of the examples that you need to know for your OCR specification. So what we can see now is that the starch, because of the chloride ion being present in the amylase, the starch now fits perfectly into the active site of that amylase enzyme. So we don't just have inorganic cofactors, we also have organic cofactors, and these organic cofactors are actually derived from your vitamins. So you need to take in your vitamins in your diet to ensure that you get these non-protein helpers, which are the organic kind, and we call them coenzymes. So these non-protein cofactors, these non-protein helpers, are actually called coenzymes because they are organic in nature. Now, there are a few that you need to know, and you will learn more about what they do when you do Module 5, but vitamin B3 is needed in your diet so that you can take in the NAD, which is a coenzyme necessary for respiration, and NADP, which is a coenzyme that is necessary for photosynthesis. So both of these coenzymes are needed, and they are derived from vitamin B3. FAD is another coenzyme that you need for respiration, and that comes from having vitamin B2 in your diet. And coenzyme A is another example of a coenzyme needed for respiration, and one that comes from vitamin B5. So it's important that you note that the inorganic, the mineral ions, cofactors are necessary, but so are organic cofactors. And they come from different places. The organic ones have to come from your diet and from the vitamins that you take into your diet. Now, we've talked about the amylase, which is a form of carbohydrates, an enzyme that breaks down saccharides. So we've talked about amylase, which is an enzyme that breaks down carbohydrates, i.e. saccharides. Now what I want to talk about is protease. So a protease, ase means enzyme, prote tells you what it's going to be working on. So a protease is a type of enzyme that's going to digest proteins into smaller peptides. So we're going to break those peptide bonds. So if we take, for example, trypsin, one of the examples you need to know, trypsin will break down proteins into smaller peptides. Now, smaller peptides then will be broken down into amino acids by another type of enzyme called endopeptidase. Now, you need to understand that the proteins are broken down into these smaller peptides first. So it's not a case of the trypsin breaks the protein straight down into amino acids. The trypsin breaks the peptide bonds, making smaller molecules, and those smaller molecules can then be digested by other enzymes. So there are lots of enzymes that you will encounter, but the key thing is that there's some you have to know. Protease is the generic term that we use for any of these: trypsin, endopeptidase, pepsin. If it is an enzyme that breaks down a protein, it is called a protease. And if it is an enzyme that's going to break down a lipid, i.e. lipase is going to break down triglycerides by breaking the ester bonds, leaving us with our monomers, which is the glycerol and 3 fatty acids. So it's important that we remember, if something says ase at the end, ASE, that is indicating that it is an enzyme. And if it is telling you the type of enzyme it is, you look at the prefix. So protease is an enzyme that breaks down proteins. Lipase is an enzyme that breaks down lipids. Decarboxylase is an enzyme which is going to remove carbon dioxide. Dehydrogenase is an enzyme that's going to remove hydrogens. So with biology, and especially with enzymes, it's important that you use the ase, which is the suffix that's telling you that that is an enzyme, and the prefix normally indicates what that enzyme is going to do. So a lot of the time when it comes to enzymes in your exam questions, they will give you something you've never heard of before, and they expect you to be able to apply that knowledge to the context of which they've given you. So I hope you've liked this video, and if you have, then please do click on the like button and subscribe to my channel. Also, if you haven't done so already, do check out my revision platform, www.aiqchat.com.